Hi, everyone. Thanks for joining. My name is Jason. I'm with MolecularCloud, GenScript, and also with me is Cindy, my colleague. She's a senior technical account manager at GenScript. And so a little bit about what GenScript is, if you're not familiar with us. We are a contract research organization. We do pretty much everything from gene synthesis to antibodies and pretty much an assay and pretty much your whole one-stop shop for life science reagents. And as many of you know, GenScript is a sponsor of iGEM, and we have been for 11 years. And this year we are sponsoring 6 iGEM teams. So far, we have only picked out one winner. One winner per month is selected for $3,000. It is $2,000 in cash and $1,000 in vouchers to GenScript products and services. Additionally, to other iGEM teams, we're going to be giving out 20% discounts to select gene synthesis services. So like your gene synthesis, your subcloning, and your express cloning to like put a gene into an expression vector. All of those, you're going to be 20% off for all iGEM teams. And yeah, we also have a bioinformatics toolbox that could help you to Hello from France. Hi, this is New Jersey. Nice to see you here. Yeah, we are going to have a 20% off discount to all iGEM teams for the gene synthesis and cloning services. And this sponsorship for the $3,000 sponsorship for 6 teams, we are doing that through MolecularCloud. And so MolecularCloud is a free platform sponsored by GenScript. And it is a permanent plasmid storage solution for labs. Like a lot of labs have plasmids just in freezers, and like ice is caking all over them. So you could store the plasmids with MolecularCloud, and whenever you need some, we can just send you some very high quality purified plasmid that you could just transfect directly into mammalian cells, because it would be industrial grade. We would remove the endotoxins. And so we do that for free, and we also help you distribute it. So if you have a plasmid that you created and you want to share it with people because you think it might be useful to someone, then you can donate it to MolecularCloud, or deposit it, not really donate it. It's because you would remain the IP holder. We won't take any intellectual property from you. We just transfer, we just facilitate the distribution of the plasmid. Say you wanted to share it to someone in Japan or France, and you're in the United States, but you don't want to do like all the shipping and all that, then you could just send it to us, and we will process the documents with that person to have that plasmid sent to them. But you would keep all of the intellectual property for your plasmids. And we also have been one of the main suppliers of COVID-19 plasmids, genes, peptides, and proteins. We have quite a few of these plasmids and proteins for sale on our website. And in addition to that, what we like to do is to promote science and the research of our active members, who we call cloud scientists. And these cloud scientists are people who are researchers, and they're publishing things or talking about science communicators, and they want to work with us to promote their science. So when you publish something, you could have your science, your research paper, send it to us, and we could promote it through email blasts, and we could go to different trade shows with it, or social media engagements with it. Especially if it's very interesting research, we can help to promote it. And so, and so our website now also has a timeline, and this is all about MolecularCloud. So the MolecularCloud has a timeline now where people can share things. It's similar to like a mix between Facebook and LinkedIn, kind of, when you look at it. So it's like you share articles and medias and like things that are really interesting, especially things pertaining to iGEM. I would personally use our Instagram accounts and our Twitter accounts to tweet them out. A lot of iGEM teams have posted about their projects, and I would share them on our social medias because a lot of them were very well written and interesting. And so, yeah, a little bit about the before I get into the sponsorship, I want to show the timeline. So one second, I'm going to share my screen. Does anybody have any questions about what MolecularCloud is at this point? I got something in the chat. Oh, hi from France. Okay, and so I'm going to pull up our website and I'm going to share the screen. Okay. Let me just get that. Always good to have as many screens as possible. Okay, and so it says everyone can see my screen. All right, so this is our page on iGEM. I don't know if you guys can see my mouse going everywhere, but that's me and at our 2019 iGEM Giant Jamboree in Boston. And so this is what we're doing this year. We have the sponsorship opportunity for 5 more teams. At the end of every month, we pick another team, and it's a voting contest. So I'm going to let's look at the sponsorship competition. Okay, and so basically this is our sponsorship competition. So for any iGEM teams who were looking for funding, you can enter the sponsorship competition. We still have 5 more slots. And so basically here's what it is. And well, here's the reward. It's $2,000 cash and $1,000 in GenScript products and services. So if you were to win the competition, you would be sent a check for this and you'd get a voucher for the $1,000 products and services voucher. And so it's a voting competition. And so what you want to do is you'd want to have your team, have one person from your team create an account on MolecularCloud, and then have them create a iGEM team page. And so we have one winner. So let me show you their iGEM team page. This is iGEM Rum from Puerto Rico. They won the sponsorship competition for April, and congrats to them. I wonder if any of them are here now. Yeah, so you'd upload a picture of your team or your team logo, and you would put in your description, your members, and the objective of the sponsorship is to collect votes. And so you would do this by having people vote for your team. You can share on social media, or you can just email people the link to vote for your team. And so the way we keep track of votes by registrants. So you have to vote by registering an account, and this is to prevent people from voting multiple times. And also you can't use fake accounts. If there's like too many fake accounts, we can't count those votes. So like asdiff at asdiff.asdiff, you can't count that vote. Yeah, that's the sponsorship competition, and we still have 5 left. So if your team has any interest in winning this sponsorship, you can go onto MolecularCloud and create an account and create an iGEM team page for your team and start collecting votes. Right now, we have we have about 42 teams right now competing. But if you wanted to get into the top 6, you pretty much just need to get 300 votes. And you could get into the top 6, and maybe one of those would beat them. But be careful. This team from India, they're doing really good. Actually, 2 teams from India are doing really good. They're very good at getting votes. Yeah. So yeah, that's our sponsorship competition. But we also help you get votes. Like I said earlier, we could we have a timeline, and if you were to tweet out, or not tweet out, write a article on our timeline, like we have a little create post, pretty much anyone can do that. That's my username. Yeah, you just create a post, and anyone with an account can post directly to this webpage. And if you write a nice article and we like it, we can tweet it out over our social medias and like encourage people to vote for your team, especially if it's something about iGEM, then we can absolutely do that. Yeah, So that is our sponsorship competition. As I had to look away when I was talking about that, but let's see, where am I? Here I am. Okay, yeah. Does anybody have any questions about the iGEM competition? If not, I can go into our toolbox, which are the bioinformatics tools that we're going to be offering to iGEM teams for free, which are going to be things like how to design a gRNA for your CRISPR experiments, or like a sequencing primer design. So yeah, we have all of this hosted on our sponsor page, and you can quickly get to all of the things for the sponsorship. So like if you wanted to get a quote for like some gene synthesis with the 20% discount, you can click this button here or learn more about the competition here. And yeah, so let's go into the bioinformatics toolbox. And so this is going to be some tools that you can use to help design sequences and gRNAs and oligos for your project. And so we actually have a lot. We have stuff all the way to like neoantigen peptides. If anyone's interested in like personalized cancer vaccines, those are the type of things that, oh, we have chats. I didn't see them. Where are they? Okay, wait. There we go. Oh, okay. Oh, no, I'm sorry, guys. I didn't have this pulled up, but the plasmid sharing service, is it paid? No, the plasmid sharing service is free. No, everything for MolecularCloud is free except for like when you want to buy a plasmid. So like we have, so for someone to donate or not donate, I keep using the word donate, deposit a plasmid with this, it's free. But for the next person who wants to take it out, say they want like, we have a CRISPR plasmid that someone used and it was really popular, then it would be $65 for a plasmid that was shared. And we would make sure that the original depositor has the intellectual property for it. But to deposit is free. And anything about MolecularCloud account is free. It's not a paid service. It's just a tool to promote your science and also, promote science and also deposit plasmids. And no, not actually. A lot of the iGEM teams right now are not doing lab work. I've seen that a lot of people are affected by the pandemic. So not everyone is currently doing lab work for the sponsorship, but you do have to be a registered iGEM team to win the sponsorship. And Kaylee, thank you. Yeah, and if you didn't win the first month, you're still in it for all of the other months. And the votes don't go away. You don't have to have the person vote for you again. Realize that a lot of people would maybe not want to vote for someone 5-6 times. So yeah, we just keep all the votes. And you're still in it, and you didn't lose your votes. And Ravi, does the contributing lab get paid for the distributed plasmids? Is this, no, you don't get paid for distributing the plasmids. It's more of something if you're willing to share. And if you were to, it is similar to iGEM. Yeah, it's similar to iGEM in that iGEM would send you a backstab, but we would send you the plasmid DNA in a tube. So it would already be extracted for you. And so yeah, sure, I can talk a little bit about depositing the plasmid. Yeah, depositing the plasmid is pretty easy. Basically what you'll do with us is you'll get in touch with us by email. you can reach the MolecularCloud team at plasmid@genscripts.com. And we have a couple of documents that need to be gone through when you first start. So like you're depositing your first plasmid, we need to make sure that your intellectual property is protected for anything that you send to us. So we have a material transfer agreement that we will that will work with you on, and also the anyone who is collecting a taking a plasmid out, they need to have some documents as well. This doesn't apply to the COVID-19 plasmids though. Those ones are just, they're just for sale. Yeah, but as to receive your own plasmid, well, it'd be rather unfortunate if you didn't have any of your own plasmid, but no, I don't think it would be free. We would still need to pay our lab people to do the prep on it and all that. But that's something, if you have you should get in touch with sales about that. If like you have a plasmid that you deposited and you wanted to have another, you wanted like 100 nanograms or micrograms of it, then maybe you should just check with your sales people, because if it's your plasmid, maybe they'll do something for you with your help, the MolecularCloud team. Yeah. Oh, we have someone who raised a hand, but I think they put it back down because I can't see it anymore. But if you have a question, why don't you just put it into the chat, because I am actually kind of, yeah, just trying to have everything on chat. If nobody has any other questions, and then I'll just get into a little bit about the design tools. If anyone has any questions about the plasmid depositing or the sponsorship competition, let me know.
Jason, I saw a new question. Not sure if you answered this already. Someone asked if MolecularCloud is like treat form for biologists, just like Amazon. I think it's different from Amazon. I think you.
Yeah, it is different from Amazon. It is definitely targeting. It's definitely for people in biology, life science, maybe biochemistry, or yeah, anyone dealing with any like bacteria or like cell lines or proteins, DNA, biotechnology. It's really so like the broad biotechnology is MolecularCloud, and it is I can't say it's like Amazon because Amazon sells literally everything on the planet, and especially right now. But Amazon doesn't sell plasmid DNA, so no, we're not like Amazon. Yeah, and I guess Madeline, are the bioinformatics tools free? Yes, they're all free. And if you go to our sponsorship page on, where are we? Our sponsorship page. Let me pull that up again. Okay, our sponsorship page. If you were to go to our sponsorship page, then you can go to our bioinformatics toolbox here, which is our, these are just like some of them, but we have a lot of them. So if you go, you just click on use toolbox, and we have all these tools for you to use. Yeah, but they are free. And how much international usage is there? Could you comment? Do you have plasmids deposited from all over the world? Does MTA work seamlessly? Europe, India, China? What are the typical timelines, say to India? Yes, we are global, and we do have depositors from China. We have depositors from the US, and we do have depositors from India. Timelines is like, in terms of timelines, do you mean like shipping? Because shipping is kind of a difficult question to answer right now. Our facilities are fully equipped to ship anything out that we need to ship out, but the shipping carriers are having issues keeping up, because everything's everything's pretty much being shipped right now all over the world. Nobody's really going to, well, lots of people are still going to Walmart, unfortunately. But, typical timelines to India. I think that it takes like 5 days to ship to India. But it should only take a couple extra days to have the plasmid purified and extracted. So I'd say like maybe a little over about a week to get to India from the US, because we ship everything from New Jersey, USA. And Europe is probably a little bit faster because it's not so far away. And any restrictions on the types of plasmid that can be submitted? No, actually, it doesn't even have to be a plasmid. You can actually, even if you don't have the physical plasmid that you can donate, not donate, I'm sorry, I keep saying donate, it's deposit. If you deposit, you could actually just deposit like a gene fragment. Say you have like a 1000 base pair coding like a really interesting gene, but you don't like have like that gene anymore, but you know it's really nice, so you can share the digital sequence with us. And yeah. And Gal, you have a question?
Jason, I saw Yu Yang raised hand. So how does this work? How does this work on the Zoom? So they can speak or something?
I guess, yeah, I allowed to talk. So actually one of my first times using Zoom. So especially as a presenter, usually on the other end. Gal, if I'm gonna let you talk, but if you don't, yeah.
Yeah, so I want to know that, how about the copyright? Say like I invented or created some plasmid and submitted to the platform, and the copyright is like what?
Yeah, so if you were to share a plasmid to us, then all of the intellectual property of that plasmid is still yours. We don't own any of it. You own it. We just, you can go to, when you deposit a plasmid to MolecularCloud as a cloud scientist, well, whenever you deposit a plasmid, you become a cloud scientist. And then you could go into your account and switch on and off which plasmids you want shared and which ones you don't want shared. And so the ones that you have switched on shared, anyone can go onto the website and see your plasmid and order it when they have an account. But that doesn't make that plasmid theirs. Let's say that you made a really nice plasmid and you shared it with MolecularCloud, and somebody sees it and they says, oh, I can make money with this, but they can't. They can't do that because this is only for research purposes. They don't own the intellectual property. If they do that, then this is only for research purposes, not for commercialization. So this is more for like academic people to use the plasmids, not really commercial. So
all right, yeah, I got it. Thanks.
Okay. Yep. Okay. Yeah. So I'll go into the Nemanja. How are we doing right now? Or do I have to?
All good from our
Okay.
side.
Cool. All right. Yeah. So if we have a little bit more time, I'd like to go into the toolbox.
Sure.
Okay, cool. Then I'm just going to go over a few of them because some of them have their own tutorials, like the construct design tool. It's like a plasmid editor, and if you go to the toolbox and go in there, you'll see what it's like. I'll just click on it so you can see it. I got to sign in first. Yeah. So so if you have a MolecularCloud account, it's free to sign up. You just give the name and email and your school, and you should be able to get in. And then, yeah, there's a tutorial for how to use this. So I'm not going to go into that one, but it's like it's a drag and drop plasmid editor. So you go into the left panel and like you choose a plasmid, and then you look for ORF clones, and you can drag and drop them into between restriction sites. And if there's like, oh, this plasmid, you put it between NDE1 and or NCO1, and now it has a frame shift, and it'll tell you about those, and you can correct them just by clicking. And then you can look at your sequence when you're done, and if you want to, you can order it from GenScript as well. Yeah, so let's go into the GenCRISPR gRNA design tool. And so here's our gRNA design tool. And so let's just say we're just going to test it with one single gene. And let's say you're doing some CRISPR experiment, and you want to have it cut a specific gene, and you just want to like either cut this gene, you want to put a mutation in there, or you want to just you want to pop it out, or you want to replace it with something. You want to cut that specific gene with a CRISPR. You can do that. And so one really popular one right now is the ACE2 receptor gene in humans. So if you wanted to use this, you'll have to find out the gene ID. So easy way to do that is to go to NCBI. I'm just going to, I have all my important hyperlinks saved. So you go to NCBI. Yeah, there we go. NCBI. And you'll go into the databases, and you'll go into genes. And you're going to want to look up ACE2. Or it could be any gene that you're looking for. And so, yeah, here's our ACE2 gene. Okay, the angiotensin 1 converting enzyme 2. And so let's say you wanted to make a double-stranded break in the ACE2 gene. You'd grab the sequence from this page here, and for sake of time, I already copied the sequence onto my Word document, but you can find any of that there. The gene ID is going to be right underneath the bolded name here. So you see this is the gene ID for ACE2 is 59272. So you wanted to do that one. You want to make a cut in the ACE2 gene. So you're going to put 59272 in here, and you can select the number of gRNAs that it designs for you. Okay, so let's just do 3. 3 is pretty standard. Like you really don't need to do 5, hopefully. And I just need to sign in. Hang on, let's just. Okay, hang on. Let me just sign into my GenScript account, so it'll show you what it does. Okay. Bioinformatics tool, GenCRISPR. Okay, I'm signed in now. And so we submit, and it just takes a little bit because it's going to process, and it will spit out some gRNA sequences at you. And so here are some little details about these gRNAs. And so these are sequences that are not also, so you're gonna want to pick the one with the highest overall score, and the score is determined by an algorithm, which is proprietary, but we do keep the, what you want to know is that the highest score is the best one. So it honestly looks like this is probably the one you'd want to go with. You might also want to try some others, but since these ones aren't as high as the 90, like there's a pretty big difference here. You might want to be very careful with just choosing one of these. This one might cut in 2 places, but yeah. So I would go with this one. And then you can take that and gRNA and use it in a CRISPR construct, and then you'll have a construct that cuts the ACE2 gene in your host. Yeah. So that's the GenCRISPR tool. Does anyone have any questions about the GenCRISPR tool? Oh, I got one for Q&A. I'm wondering if MolecularCloud. Oh yes. Okay, we just got that one. And okay. Sorry, we have the chats and we have the Q&A. So sometimes I don't get everything. Okay. And so do you ship genes expression ready, already cloned into plasmid of any plasmid of choice? So that's actually going to be something that we need GenScript's help for. And this is why we are sponsored by GenScript. It's a platform by GenScript. And so if you can get a gene, what you can do is you can get this, if you were to go to MolecularCloud, and let's just say we want, actually let's go to the COVID-19 plasmids. So we have lots of COVID-19 plasmids. So like this is the ORF7A protein, which is in COVID-19, the SARS-CoV-2. And this one comes in pcDNA 3.1 with a P2A and EGFP. So it would, it's going to come in a mammalian expression vector with a self-cleaving enzyme between the ORF and the GFP. So it's going to glow, but it's also going to cut off the GFP in the host. So the protein won't be attached to GFP if you wanted to purify it or something, then you wouldn't have the GFP attached to it. Yeah, and this one you could add this to your cart, and the quotation process is handled entirely by GenScript. So you would get the quote from GenScript, not MolecularCloud. The quotes for like any of these things would come from GenScript. Then you would you'd be able to add on services like subcloning into a different expression vector. Like say you wanted it in you didn't want it in pcDNA 3.1, you wanted it in like pCMV, or like you have your own vector that you like to use, then you could do that. You could ask GenScript to do the subcloning, and since you're an iGEM, since you're in an iGEM team, you'd get 20% off on the subcloning. But yes, we do have a lot of genes that are expression ready. You can go up to the top here, and you could see all of the. You could Google, or not Google. You could search. Google and search are pretty much synonyms at this point. Nobody uses Bing, and yeah, you could search for them, and yeah. So some of them are expression ready. Some of them need to be cloned. Should we pay by ourselves for those data? Which data? I might have missed what you were saying. If you're referring to what time is it? 9:34. You said at 9:33. Then you were probably talking about something in here, or the GenCRISPR. Okay, so the COVID-19 one. So these ones, which data? Would you, could you raise your hand so I could let you speak and you could answer the, all right, cool. Okay, yeah. I've unmuted.
Yeah, because there's a button called add to cart, and before that there is the price, like $140. So I want to ask, should we pay for that, or these informations are sponsored by GenScript?
Okay, yeah, good question. So these are, all of the stuff from this page actually are like actual like products that we're selling. So these ones are going to be things that you would have to pay for unless you win the sponsorship. Then you'd get quite a bit of play money to get things like this. Yeah, so they actually range in price pretty dramatically depending on how large the gene is. Like we have like the spike protein too. But yeah, it is $140. If you are working with with COVID-19 or like genes related to COVID-19, I think it's always a good idea to check with your sales. So if you get a quote, you'd add it to cart and then you'd be given a quote first. You don't just immediately go to checkout. I mean, I think you actually could, but I would suggest to just get a quote and meet your salespeople, because a lot of the time they are willing to work with you to meet your budget for things like that. If talking about like plasmids, like a CRISPR plasmid, we just go up here, find a CRISPR plasmid. These are going to just be like, okay, let's see if this one. Okay, so like this one right here is some plasmid that was deposited by one of our cloud scientists, Sheng Yang, in Shanghai Institutes for Biological Sciences. And yeah, it's a CRISPR helper plasmid. And it's $65, but you'd have to sign the material transfer agreement for it. But anything with a price is a price. They're not free.
Okay, I appreciate that.
Mm-hmm. Okay. All right, yeah. So yeah, that was the gRNA design tool. Another thing that you might be doing a lot is, well, I used to be an iGEMer back in like 2014. I was doing iGEM. And one of the things that I was doing was PCR primers. And so let's say you were just, you needed to do some PCR, and I just need to grab any sequence to show you how this works. So I'm just going to grab the ACE2 sequence because I'm really interested in the ACE2 gene right now. And so you just put the ACE2 sequence in here, and it would you pick primers. And actually, the one that you probably want to do is sequencing, because actual primer design is actual primer design. You just pick the one on the left and the right. Then yeah. So the sequencing primer design tool is a little bit different, where you just put this in here and you would want to pick the distance between the two primers up here at the top. You can change the distance between the primers, and you can also work with your melting temperature. And yeah, so yeah, you put your sequence in here and you can design primer, and it will spit out a bunch of primers for you. And so when I actually started at GenScript, I was one of the sequencing technical account managers. So I mainly worked with the sequencing service line. You don't actually need to use every primer that is suggested to you. You pretty much just want to get coverage for the region. So like you want to find like 400 to 1, and then you could like flip. You could like, the read will give you like 500 base pairs, 400 base pairs. You want to flip it over, and then you can just use that, and then use the 401 to 800. And that should give you some coverage between the two, and you should be able to get like your whole protein when you use this one, and it should be done. So you pretty much just want to start with one reverse and then use these. You don't need to use all of them unless like you really are not, like you have like some heavily mutated piece of DNA and you just really want to be sure about mutation coverage. Then I would suggest to use all of them, but like you just want to make sure it's like not messed up, because PCR will have 1 in 3,000 base pair mutation. Then yeah, so that's the sequencing primer design tool, and we have quite a few others, and they're all on our list here. like we have this Wolf P sort will show you where the protein subcellular location is. Like say you're doing some GFP in whatever, you can check and see where GFP has expressed subcellularly in other research papers, and you'll know where to look for your gene, which could be pretty helpful, depending on what your research project is. But we have a lot, and I do suggest that everyone, if you have the time, check out these tools. Maybe we'll have something in here that can help you with your research. You maybe instead of crunching numbers and looking at staring down papers all day, you can just use one of these calculators to help you out. And yeah, I think, am I missing anything in the chat? Okay. Does anyone have any questions at this time about the sponsorship or the anything else? And if not, then I think I'm going to sign off then. All right, fellas, thank you for coming to the GenScript MolecularCloud the virtual trade show thing. iGEM opening weekend. Yeah. Good morning. Thanks, everyone.
Thank you, Jason, for presenting.
Of course. Thanks for having me.
And thanks everyone for joining. We will take a short break and then we will be back in 15 to 20 minutes for the iGEM HQ welcome to the festival. Thank you all. See you later.
Thanks, Jason.
Thank you.